Good morning, ladies and gentlemen. Thank you very much. I've been absolutely thrilled with this Ed Forum and the effort everybody's put into it and your response and support, and it's been so much fun. We have a great opportunity this morning. Pope Treon is going to lay the groundwork for two young people that the IWMF has sponsored for research. We want to just explain to you how rigorous, how careful, how important it is us to fund the research, but not fund research, fund the right research to make a difference for clinical well-being of WM patients. So let's welcome Steve Treon.
Thank you, Paul. I still have not seen the white smoke, and there is no ring. But it's really a pleasure to be here with all of you. And I got to say, it's particularly exciting, this session. We're going to get a chance to meet the young investigators who have gone on to receive the Robert A. Kyle Career Development Award. And this, I have to say, is exciting because it represents the next generation of leadership in this disease. And this is why we've invested in them by creating the Career Development Awards. I had the opportunity to chair the first several committees that selected our awardees. We've sort of rotated that chairmanship, but it came back to me this past year. And I got to say, you're going to meet 4 of our Career Development awardees, but the 4 that got selected this year are also incredibly talented and will really bring, much new light to Waldenström. So this is really a great investment. And again, I encourage you, support the IWMF because these are the awards that help us get people off the ground. It helped me when I was actually one of the first awardees of a Career Development Award way back, I'm going to say almost 30 years ago. But it does allow us to be able to have our young investigators focus on this disease, particularly at this crucial moment in their careers. And especially now when you're seeing all the federal funding beginning to, become curtailed. So again, thank you for supporting this endeavor and continue to do so. Now, we have actually with us 2 live Robert A. Kyle Career Development awardees. And we have 2 virtuals. One's actually going to join us by Zoom. The other actually had to pre-record their presentation because he's actually actively traveling. So we're going to start off with Philip Garbitz. Philip, I've had the opportunity to get to know. He is based at Dana-Farber Cancer Institute. He works very closely with Ruben Carrasco, who is truly a hero to all of us that work in this myeloma Waldenström's field because he's helped us understand the basic genomics of the disease by creating these transgenic mice. These are mice that have been engineered to be able to have the mutations that actually drive disease. And in particular here, Philip's going to tell us about his work with MYD88. And one of the things we've learned is that just putting the MYD88 mutation into a mouse doesn't create Waldenström's. And we're going to hear from Philip where his work is taking us now to understand how other cooperating mutations actually play also a fundamental role in helping MYD88 do what it does. And of course, ultimately, we want to learn about how we're going to stop all this. And these models that Philip is working on are quite illuminating. So, Philip, why don't you come up, give us your presentation, and then we probably will have a few questions for you. So please welcome Philip.
Thanks, Steve. Good morning, everybody. Okay. Good morning. So today I wanted to navigate the deep with you. So I wanted to answer the question, very difficult question. Can mouse models help us map the biology of WM? And first, I wanted to give an introduction. There's a very standard model that we all know when studying the pathogenesis of Waldenström's. We've learned from studying patient samples, cell lines, that there is this Waldenström initiating cell, which is a B cell that's experiencing an antigen response. It's stimulated by the immune system to proliferate. And then the B cell is trying to differentiate, which is to change to its final form, which is changing into either a plasma cell, which secretes antibodies, including IgM, or a memory B cell. And for some reason, there's a blockade at some point of this differentiation pathway, most likely a blockade caused by one of the mutations that happens in this proliferating B cell, which renders those B cells not able to assume their final form. And they start to proliferate, they keep proliferating, but in a very aberrant way. And this is, as I say, this is a model because we have really never seen that in your samples. When the patient with Waldenstrom comes into our office, they already have all the symptoms, they already have the disease that has developed in the bone marrow. So we're not really sure what happened, 5, 10, 15 years before that visit, before that first visit. And what can help us understanding this process is mouse models. So mice don't usually get Waldenstrom. Actually, it has not been reported that mice can suffer from Waldenstrom, but they age pretty fast. They live up to 2 years, and they get many age-related diseases after 6 or 12 months since their birth. So they are the perfect model to study age-related cancers such as Waldenstrom. And this makes it also possible for us to use those mice to track this disease almost in real time to see what is happening before the disease fully develops. And to create such a mouse model, we really need to know about the genomic complexity of human Waldenstrom. And this is where Steve's and Zach's work comes into play, because they've been able to tell us that up to 90% of Waldenstrom patients have this MYD88 mutation. But the second most common alteration in this patient group is what we call deletion 6q. And this is not really a mutation per se. This is a removal or loss of a huge chunk of DNA on our chromosome 6, the long arm, which is the Q. And this is causing maybe dozens or hundreds of genes to be lost. So they're completely not functional, they're not there. And many of those genes, what we found is they're what's called the tumor suppressor. So they're genes that are supposed to keep our B cells in check so that they don't proliferate, they don't cause cancer. And for some reason, up to 50% of patients, Waldenstrom patients, don't have those genes at all. So using this knowledge and using Steve's input, we created a very sophisticated genetic model. And I'm not going to go into the details how this model works, but it's really, I think I'm one of the few people in the world that really understand what's going on, because I'm the postdoc on this project. But what's going on here is that the chromosome 6q, the human chromosome 6q, has a very, very similar region to the murine chromosome 10q. And so we look at the murine 10q, we found the sites where the chromosome usually breaks. We inserted some special genetic sequences, which then gets this whole 10q removed, but only in B cells, which is the cell that initiates Waldenstrom. And these are the results of our studies. We've been observing those mice already for many, many years. And on the left here, you can see the healthy murine bone marrow. And what you see here is basically a slide stained with IgM. So we're looking for IgM positive plasmacytic cells in the bone marrow. On the left, you can see just 2 or 3 plasma cells, which is the normal state. But on the right, you can see 2 pictures of bone marrow from mice with 10q deletion or 10q deletion with MYD88 mutation expressed in different kinds of B cells. And you can see that the bone marrow is infiltrated with those lymphoplasmacytic cells, which are so characteristic of Waldenstrom. So actually, my boss is a pathologist. I showed him this slice, and he said, well, Philip, I think you found a model of Waldenstrom. This looks exactly like the human condition. So what are we doing with those mice? As I said, we can track Waldenstrom development almost in real time. So we're now collecting tissues from those mice at different time points. And we can see using high throughput sequencing technologies that those cells start proliferating, and those proliferating clonal, what we call cells, part of the cancer clone, are depicted on the right here with this red-black color. We can extract information from those cells and then look at the cellular program and tell exactly what went wrong, why these cells could not differentiate, and why are they proliferating and causing this disease. Other methods that we're using in the lab, we're trying to make this project also a little bit translational. We are trying to take the bone marrow from those Waldenstrom-like diseased mice and then incubate in vitro or test in vivo whether they exhibit drug sensitivity similar to the one that's observed in Waldenstrom. So here is just a couple of results. Literally from 2 weeks ago, I was able to extract bone marrow from one of the mice, and I tested the sensitivity towards different standards of care in Waldenstrom. And you can see on the left here, I grouped some of the drugs that work really well in most of the patients. So we got venetoclax, ibrutinib, dexamethasone, and the Waldenstrom cells from those mice showed really high sensitivity. So they were killed very effectively by those drugs. When it comes to moderate efficacy drugs such as proteasome inhibitors, you can see here bortezomib, for example, those cells were still sensitive, kind of sensitive in the middle. And you can really appreciate the result when you get some kind of negative result that you also expect. So on the right here, you can see a drug that really did not show any efficacy in our in vitro cultured mouse model. And this is an AKT inhibitor, the best in its class, current best of class. And the cells did not really care about this AKT inhibitor. And what's interesting is that 14 years ago, there has been a trial in Waldenstrom patients which tested a different AKT inhibitor, and also the patients did not respond almost at all to this inhibitor. So you can imagine here that this mouse model can be used as a tool to study some drugs or drug combination pre-clinically. So before we start a clinical trial, we can first look at the response in mice, and mice are basically coming in unlimited numbers. We can generate a cohort of 50 mice, 100 mice, test different drug combinations, and then see whether the cells die a little bit or do not care at all. And the other thing that we can do is test toxicity, because you saw some results from trials that were terminated because of toxicity. And when you treat the mice with certain drug combinations, you can also see this toxicity in the mouse models. So with that, I wanted to thank IWMF, first of all, for inviting me here, for supporting our research. It's very important to acknowledge the foundation that's given me so much hope to move this project forward. And I also wanted to thank my lab, Carrasco Lab, as well as the Bing Center from Dana-Farber. Thank you so much.
Yep. We're going to take questions and do our interview at the end when we have all our panelists together. It gives me great pleasure to next invite David Cortes dos Santos to give us a presentation. David is also at Dana-Farber Cancer Institute. He's been very excited about the field of plasma cell malignancies, myeloma, as well as Waldenstrom's. And he's a trained physician in Germany at the University of Munich, which is a true center of excellence in Germany. But while he's been at Dana-Farber, he has been working with Irene Gabriel and trying to understand how precursor conditions, like we've heard about IgM-MGUS, the precursor for Waldenstrom's, how molecularly these entities begin to evolve towards Waldenstrom's, both asymptomatic and then symptomatic. And so we've learned a lot from you, David, from the work that you've been doing in myeloma. I'm so happy that we've been able to recruit your expertise to Waldenstrom's, and very excited to hear your presentation today. So please, let's all give David a warm welcome.
All right. thank you so much for the invitation. I'm very happy to be here. And I think we had some German accent imitation on Friday. Now you get the real German accent. So the title of this talk is Mapping Smoldering WM, and you look inside the bone marrow. And I want to start with, what is the big question of our lab? So Stephen just said it. Like in our lab, we were like in the lab of Dr. Gabriel. We're looking into why do some patients who have a precursor condition, for example, smoldering WM or smoldering myeloma, some of these patients stay well for years, while others, they kind of a little bit sooner develop active WM or active myeloma. And we do know that, especially in smoldering WM, we do track age, IgM levels, genetics, and some of these factors in patients who have smoldering WM, they affect the progression, right? So we know that some of these factors are actually somehow contributing to progression rate. However, what we want to ask in this topic, or in this research that I'm presenting now, is like, can we find missing clues? Because obviously we cannot, like, for certain tell a smoldering WM patient you're going to progress at this rate, or like at some point, and you're going to develop symptoms. So the question is, can we define or refine the model and our prognostication of this even further? Why would we want to do this? Maybe we want to get early help for higher risk patients, or we want to spare lower risk patients from extra tests and worry, right? So how do we actually look at Waldenstrom cells or WM cells? And how do we peek inside? And I think probably a lot of you had a bone marrow puncture already. And, you know, it's not the nicest thing to get as a patient. But it's kind of necessary, right? Because this is the way we identify the Waldenstrom cells. And I'm gonna refer back to the session of Maury Gertz from Friday. He talked all about the garden, right? The garden and the weeds are like the Waldenstrom cells. So everything is going to be around this in this talk. So if you remember this was a nice image. So I'm gonna pick this up at some points. When we look at bone marrow punctures, there are actually two things that we do with them. One, we call this a bone marrow aspirate, and that's like this quick suction of liquid marrow, like the bone marrow blood, right? And then the other thing that we do is actually a bone marrow core biopsy. Well, this is like the part of the bone marrow puncture, which is not very nice because kind of sometimes it hurts, right, and tweaks. And this is when we take like a tiny solid plaque of bone and marrow. But this is really interesting for us because it shows us, we do the slides, like what Philip just showed in his slides. We have a slide of the bone marrow. We can actually look how the marrow really looks like. Whereas in the bone marrow blood, we lose the information about how cells are localized spatially in the actual tissue. So the other good thing for the bone marrow core biopsies is we can store them for years, so for decades. They're safe. They're just in a block, and we can just look at them also years after they were taken. So what do we do in usual like clinical routine? So we stain the cells. We do like, this is how a bone marrow image looks like. Is there a pointer? No, wait. And based on like how the cell looks like, what color it has, we can identify like the appearance of the cell. We can say it's this or that cell. And sometimes we can use like 2 to 3 markers in the clinical routine and say, oh, this specific cell, like if it's like more brown here in this image, this is most likely, for example, a WM cell, and we can identify them even better. However, if we only have 2 or 3 colors, right, we will not be able to look at the entire complexity of the bone marrow, so we lose a lot of information. Why do we want to look at this anyway? Because somebody could say, well, you already have like all the genome sequencing. We couldn't look at the genetic information, the DNA, or the gene expression levels. But the thing is, imagine you have a fruit salad. I always love this picture. You have a fruit salad, and you put all the fruits in the mixer, and you make a smoothie. When you get genetic information of this smoothie, you can say, well, you get like a very broad picture. You can see, well, there's a MYD88 mutation in the smoothie, right? But you don't know from which cell it actually comes. So what was done the last 10 years is you can we can now look at genetic information of single cells. So even if you have like a fruit salad, you can say, oh, there are apples in there and oranges and grapes. And now you can say, well, these apples, they have this and this genetic information. And we know that, for example, this mutation or something else comes from that specific fruit. So what does spatial technologies actually give us? In spatial technologies, we actually look at the fruit salad itself. And we can say, well, this grape on top has maybe a different genetic information than the grape on the bottom. So we can actually really look in the tissue and not only identify the cells, we can also get information about the genetic information of each cell within the tissue. So it opens a new dimension. So how do we do this in the study that we're working on? We kind of created a new camera. So instead of like these 2 colors that I showed you before, we now look at 36 different colors. The technology is called imaging mass cytometry. Forget this, not so important. But important is what we kind of can create here, and this is what we show in these images, is kind of something like a Google Maps for your bone marrow. So we can identify, when you look on the lower right, each of these colors here is a specific cell in the marrow, but now we can say it's this specific cell, and we can do this at one shot in one kind of, on one level. So we don't have to do multiple levels and different factors. We can all do this at once. And this is really great. We did this in a cohort of different patients who got a specific treatment, and it's kind of the first time that it was actually shown. When we use like high mathematical algorithms, I'm not going to go into this, we can identify specific neighborhoods in the bone marrow. So we can say, well, when we go back to the garden, right? There were roses and tulips, and like, are the roses always surrounded by other roses, or are the roses surrounded by tulips as well? Are the weeds surrounded by roses and tulips, or are the weeds only staying within themselves? And now we can identify these kind of neighborhoods, right? And then we can associate this with survival or with progression, for example, from smoldering WM. And this is what we showed in the first kind of study that we did, that it actually works and we can identify these neighborhoods. And these are just some snapshots of these neighborhoods. And when you look on the right one, you know, blood vessels which go through all of the body, they also go through the bone marrow. And when you imagine like a blood vessel as a tube, and now you cut the tube in half, this is what you can see on the lower right. So this neighborhood is kind of, the tube is made of specific cells, and if you cut it now, you can see, what you can see here is like if you would look into a tube, so we actually can identify this tube neighborhood. So what are we going to do with this now? What we are doing now is we take a lot of samples from smoldering WM patients. I screened over 150 patients, like patient samples right now. We do this analysis on all of these samples. We integrate this with clinical data and genomic data. And then we try to really take all these puzzle pieces into one model to be able to say, or to create like a personalized risk estimate for the patients. So we would love to say, or to answer the question even better, who progresses from smoldering Waldenstrom to active WM? Or we could also answer the question, who responds to treatment better or worse, or is this treatment better for that patient, or is this treatment better for another patient? And also this opens new paths to new treatments. So maybe if the weeds are always surrounded by a certain cell, which is a helper cell for the WM cell, can we modify this neighborhood, or can we modify these helper cells, maybe eradicate them so that the Waldenstrom cells do not progress or are not like supported by other cells? And with this, I want to thank every patient and family for making this research possible, and obviously also IWMF. I have to say, I actually did not get the award myself. It was Yoshinobu Konishi. I worked very closely on all of this with him together, but he was appointed a professor in Japan, so he went back to Japan now, and I took over this his research and like the grant and the study. But well, we worked on everything together in the lab, so it's actually a perfect handover. So I'm very happy to be funded by IWMF and take this in, to the next round. Thank you so much.
Okay, we're about to enter the virtual world now. So we have Tina Bagratuni, who's going to be presenting us from the University of Athens. Tina works very closely with Thanos Dimopoulos, who I think needs no introduction to this group. Thanos has been really a key, mover and shaker in the field of Waldenstrom's, having been involved in the development of drugs for over a 30-year period. But I was particularly excited when he recruited Tina to his team to be able to advance many molecular studies that have impacted Waldenstrom's. And Tina has also been the recipient of multiple awards at the International Workshop on Waldenstrom's, and she now has the Robert A. Kyle Career Development Award. So we're going to be hearing from her on a topic that is really exciting and important, and that is what happens to patients when they're on drugs like BTK inhibitors, like ibrutinib, and how do their tumor cells actually evolve? In some cases, lead to resistance. And I'm very excited to hear about her work because truly this is going to be important and impactful in the way we understand and position BTK inhibitors for treating Waldenstrom patients. So Tina, welcome. Thank you so much for being here at the forum. And we're really excited to hear about all your work.
Thank you very much. Good morning. Thank you very much, Steve, for your kind words. And yes, I'm Tina Bagratuni. I work at the University of Athens, and I've been receiving the Robert Kyle Development Award for the project entitled Tackling the Evolution of Ibrutinib-Resistant Protein in Waldenstrom. So what does resistant mean and how can proteins help to that? So resistant to therapies means that the treatment stops being effective. And this can eventually lead to the cancer growing and making these treatments less successful. So it usually develops due to different genetic alterations that happen at the DNA level within the cancer cells themselves, but it can also be significantly affected by the surrounding microenvironment, known as the tumor microenvironment. So proteomics, which is the study of proteins, have greatly helped in understanding why cancer cells become resistant to specific treatments, and the proteins themselves can play a very crucial role in how cells function and how these changes at the protein levels and the protein function give us a much better picture of what's happening inside the cells, but also in the tumor microenvironment. So everything starts at the DNA level. So we have the original DNA, which has no mutations, and then we have specific alterations. These are basically characterized by synonymous or non-synonymous mutations. And what we know to a certain extent is that these might actually have an effect to their genes too. So we might have a lower expression, even in the case of the non-synonymous or the synonymous mutation. We might have a completely normal RNA expression at the gene level, but we might also have an mRNA decay. But what we know for sure is in the case of a non-synonymous mutation, we will have an amino acid change at the protein level. This will change the protein sequence, and this might have an effect at the protein expression level or the protein function. So based on the DNA and mRNA, we got the first grant from the IWMF, the Research Roadmap Initiative grant, which basically what is trying to do, this is an ongoing research trying to examine why some patients with Waldenstrom are becoming resistant to ibrutinib by looking closely at the gene levels and the mutations in the DNA in these patients in an effort to try and understand how these might affect the resistance mechanism. So now that we have enough data, we have generated enough data, and we've found some interesting results from these studies, I felt that it would be good to actually see what happens at the protein level. And this is why I applied for the Robert Kyle Career Development Award, just to see the same exact things at the protein. So I just want to mention that mutations matter even if proteins stay the same. So this is very important to do all of the genomic studies in the same time. So the aim of this study is to take a comprehensive approach to study the proteins in patients undergoing ibrutinib therapy and hopefully help identify these processes that allow cancer cells to grow and survive, which will eventually lead to a more improved strategy for treatment for patients that are affected by this ibrutinib-resistant mechanism. So the design of this study is we have a number of patients that they are just about to start their ibrutinib therapy. These are 40 in our case, and this is called as a baseline time point. And after 6 months, we already know which patients have responded to ibrutinib and which have not responded. But we also know that some patients are more slowly responding to the treatment with ibrutinib. So for both of these cases, we do have bone marrow samples and peripheral blood at baseline and 6 months. So what we aim to do with the bone marrow samples is do what it is called a mass spec analysis on tumor cells and tumor microenvironment. And this is actually trying to see the entire range of the human protein profile, just every protein that it is expressed in the bone marrow, in the tumor cells, and the surrounding microenvironment, both at the baseline treatment and 6 months after treatment. Whereas in the case of the peripheral blood, we're going to take the plasma and we're going to do the plasma proteomics, which is by technique called the plasma proximity extension assay, which basically sees a range of 6,000 proteins. So these are basically linked to every cancer or non-cancer related disease in a very specific and sensitive way. So we're going to combine, try to do the clicker here. So we're going to combine a plasma proteomics at 6 months and baseline, and we're going to do some bioinformatic analysis and try to find the disease biomarkers which can predict which patients are going to respond to ibrutinib and which are not. Whereas in the case of the mass spec analysis, we're going to combine all of the proteomic data with our first study. This is the mutation profiling and the single cell RNA sequencing data. I'm going to combine all of this data to what is called a multi-omic approach with an aim to understand how the story goes, how changes at the DNA level can affect the RNA, with eventually affecting the protein expression analysis. So the input of the findings for the first aim, we're trying to provide the differences between the cancer cells that are responding to therapy and those that are becoming resistant to it. And by looking closely at the proteins and the gene activity in these cells, we hope to better understand how this resistance develops. And hopefully this knowledge will help doctors will find a more precise drug treatments or maybe a different combination of drug treatments which might be more effective to these patients. Whereas in the second aim, we're going to try and explore ways in finding disease biomarkers in blood samples with an effort to avoid this repeated painful bone marrow tests. So if it proves to be successful, this approach could help doctors track changes in the disease and the response to treatment using just a simple blood draw. So just a few preliminary data. We have started with the plasma proteomics. This is an ongoing study for about 6 months now. And what we try to do here is to find these features, the protein features that can best separate these two groups of patients, the responder and the non-responder. As you can see from the red circle, these are the non-responders, whereas the yellow ones are the responders. And we can see in the heat map analysis, this is just on the right side, which are the proteins that are most seen in the non-responder and the responder groups, etc. And then we can actually focus on those proteins specifically and see how they are actually linked to the disease. The other thing that we try to do here is to see what are the proteins that are targeted with ibrutinib. So we have done the data from before the treatment starts and 6 months after it starts, and we can see some differences just to know how ibrutinib actually targets these proteins. And again, we have this heat map analysis, which in this case we have focused on the circulating cytokines and chemokines. These are actually involved in the inflammatory pathway. We already know that they are linked to disease. And again, here we can see the differences among the responder patients and the non-responder patients, both at the baseline and 6 months after treatment. So the perspective of this study is a project which builds on an earlier research that we have studied the genes and the activity of the cancer cells in Waldenstrom. The goal is to combine different types of data to better understand how patients respond to ibrutinib, and hopefully these findings may help doctors to develop new treatments for patients whose cancer stops responding to ibrutinib. So as I was trying to see how to end this presentation, I felt that I should use some elements of history, and as a Greek, I had to quote something from Greek history. So this goes back to 450 BC, the Battle of Marathon. So this was a fight against among Persians and Athenians, and Greeks, as always, underpowered. They had very few men and few weapons. So they had to come up with a strategy. So instead of actually confronting the entire enemy at the fronts, they actually, with a very unnoticeable way, they tried to surround the enemy and gradually lead them to the sea. And this was actually called as a defeat for the Persians and a victory from Greeks. So taking this into the world of biology, I would say the Persians are the Waldenstrom cells, and what we're trying to do here is target the cells, of course, mitigate ibrutinibs and ibrutinib, pretibrutinib, but we can also target with other inhibitors. These are BCL2 inhibitors or BTK degraders, but we can also find other drug targets. But what it is important is that we can actually surround the cancer cells, the tumor microenvironment, and we know that the bispecific can do this by actually targeting both the cancer cells and the tumor microenvironment. But all of this is conceptualized and designed by the Waldenstrom community, the research community, and the clinical community. And most of these studies are actually with the support of IWMF. So I would like to personally thank IWMF, which has supported and has given me a great opportunity on working my research ideas. And I hope that all of these studies eventually in the long term will help patients getting a much better clinical management and a much better quality of life. So thank you very much.
So we got science and we got a valuable lesson in history today from Tina. Tina, I know you might be actually hearing us, but hang on. We're going to actually go on to see a video that was prepared by David Moreno, who will be our 4th Career Development Award recipient for presentation today. And we'll have a couple of questions for you, so hang in there. So our next presenter is David Moreno. David is also a mover and shaker. He's based at the University of Barcelona, where he's been working with Carlos Fernandez de la Rea, and truly a center of excellence when it comes to myeloma as well as Waldenstrom's. And he's made tremendous contributions in understanding basic diagnostic testing for Waldenstrom's around mid-88 CXCR4, showing us how the mutation rate for both of these genes changes, evolves as one moves from MGUS, the early stage of Waldenstrom, pre-Waldenstrom, to symptomatic disease. And these days, David is focusing on giving us a lot more insight into the genome as it evolves from the early precursor states to active Waldenstroms. Unfortunately, he's traveling right now, and we tried to be able to fit him into a Zoom, but we thought it best to actually just pre-record his presentation. So he won't be available for a Q&A, but we're going to get an insight into the work that he's doing. David also has received numerous awards, including the Young Investigator Award from the International Workshop on Waldenstroms. So, Cindy, I think it's time to play David's video.
Good morning. My name is David Moreno. I'm a hematologist currently working in the VAPS and Hospital Clinic of Barcelona. I am very grateful to have the opportunity to talk about the work I do and the project that I'm carrying out thanks to IWMF. I'm a postdoctoral fellow with the intention to become a physician scientist specialized in IgM gammopathy and Waldenstrom macroglobulinemia and be able to combine clinical work and research. So my story begins in Lima, in Peru actually, where I was born. There I studied medicine, and in 2016 I moved to Spain to continue my training on hematology. And at Hospital Clinic of Barcelona, I was very fortunate to work with mentors in this field who supported my initial steps. But my journey had also taken place in other places, actually, where I had the pleasure to work with outstanding mentors. In fact, what inspired me the most was that during my hematology fellowship, I had the opportunity to spend time at the Waldstrom Macroglobulinemia Clinic at Dana-Farber Cancer Institute. And there I was struck by how novel discoveries were actively transforming treatment strategies with the goal of improving patient care and patient survival. And this translational perspective of the disease increased my interest to further pursue research in this area. I also did other short stays, like a couple years ago in the EMBL and EBI in the UK to study the human genome, and a year ago in the Prinses Maxima Centrum in Utrecht in the Netherlands. So I have been very enthusiastic about combining clinical care and research, which was the reason I was enrolled in a doctoral program of translational medicine. And this journey allowed me to conduct an in-depth study of IgM gas and Waldenstrom macroglobulinemia. And that culminated in the successful defense of my thesis a year ago. I am now in the postdoctoral stage of my career, with a specific focus on genomics in Waldenstrom macroglobulinemia and how it can help the management of patients. This is why I see myself transitioning between the clinic and the computer lab. So all this work was supported by people who guided me in the clinic. As you can see, I am the one here smiling, and in the lab. So the project that I'm presenting now is about how analyzing individual cells from Waldenstrom macroglobulinemia patients can help to discover new biomarkers. All this started by a simple question. Can we deeply understand Waldenstrom macroglobulinemia cells so we can trace back to its origin and see how these cells evolve? So what we usually see in the clinic are, for instance, patients diagnosed with IgM gas that have very low amounts of serum IgM. And with time, some patients acquire extra genetic alterations that select groups of cells that can be the cause of symptoms, for example. And tracking these genetic alterations at single-cell resolution is essential then to understand disease progression. For instance, cells can evolve in a linear fashion, but we now know that a cell can have multiple histories during the follow-up of patients. We can think that each Waldenstrom macroglobulinemia cell is a patient. Each one of them is unique and have their own story to tell. And why is this important? Waldenstrom macroglobulinemia is composed of multiple cells, and we can see them as a mosaic, like this picture from a Gaudi work in Barcelona. Some cells can cause progression or refractoriness to treatments, but others can make a patient asymptomatic during the entire lifespan. And how can we track these cells now? Genomics has been transforming patient care for years, and the techniques used to study cancer genomes have advanced dramatically. The resolution is now so high that we can sequence the entire genome of a single cancer cell. And to do this, we are planning a series of experiments that can capture both the complete set of genetic information, which is the genome, and the messages that genes are sending out, which is the transcriptome, from each individual cell at the same time, simultaneously. We will use samples that are stored in our biobank from patients with IgM gas and WM. So the technique is called Primary Templated Directed Amplification, and it stands for PTA. And in short, what it does is it amplifies tiny amounts of DNA from a single cell with low error rates and artifacts. We can also fragment a cell to obtain intact DNA and RNA separately. So we expect to discover novel genomic biomarkers that can help us to delineate clinical outcomes. For example, how a mutation causes an increase in certain proteins that can be further used in the design of new targets in clinical trials. And given the design of the project and the use of longitudinal samples, we will also estimate when these mutations happen during the life history of a patient, with high potential in designing programs for early screening or intervention. Also, we will give answers to key questions like, do cells that have the MYD88 mutation cause progression, or only those that have MYD88 and CXCR4 mutations, or P53 or deletion 6q and other molecular biomarkers that we now know? Which genetic alterations make an individual cell to grow and expand finally? And what is the impact of those genetic alterations in each cell? We are carrying out the project now, and during these months we have been setting up the techniques and running the first experiments and analysis. And the intention in the upcoming months is to expand to more cells and samples that will answer the key questions underlying the Waldenström macroglobulinemia origin and progression. I am very grateful for the patients that give support to our research and all the people involved in the clinic and the lab. And I am truly thankful to IWMF that has given me the opportunity and the support to continue my research on Waldenström macroglobulinemia. Thank every one of you, and if you have any further questions, please do not hesitate to contact me by email. Thank you so much.
Well, we had 4 excellent presentations. What do you think, Paul?
I think it's great to have young people participating. I'm really anxious to ask a couple questions about how they're going to go in the future and where we're headed. So if you would just take a couple of minutes to say what receiving that grant meant to your career and what it meant to you personally as a young person starting as a professional science.
Of course. So I came to the US a year and a half ago, not knowing anybody other than my boss. And then I met Steve, and Steve inspired me to work on Waldenström's and take a look at the mouse models that are available at Dana-Farber to develop them further. Then I applied for the Kyle Award, received the award, and basically chose this project as my main project for the rest of my postdoc. I came to the lab, and the lab has multiple different beautiful projects. We're studying gastric cancer, liver cancer, multiple myeloma. And I said to my PI, you know what? I got this award now. I got to work on Waldenström's. You have to give the other project to somebody else because this is my main focus now. And so this has really changed my professional trajectory. I hope for the rest of my professional life. And I'm extremely, extremely grateful for this opportunity.
And personal?
And personally, so well, being postdoc is really hard. The first year of my postdoc, I was in a long-distance relationship with my wife, who's amongst you over here. Actually, happy birthday, Julia. It's her 30th birthday this weekend, so... So, I was basically living without my family for 1 year. But what this award gave me is, a family, a professional family. I was really feeling a part of this community, and I was getting support. Every time I have some kind of problem, I can always talk to Steve or Zach Hunter, and all the meetings that IWMF is organizing are just wonderful events. Actually, the last year, the meeting in Prague, I was able to invite my wife as well. So we spent some nice time together in Prague while we were long distance. So it has impacted my private life as well.
That's fantastic. It's rewarding because the IWMF is looking looking for the long term. We're looking for the short term answers. And it's great to have young people like you participating. So thank you very
Thank you.
much.
And Philip, I have to say, the work that you're doing is very important because it gives us a model, a realistic model that we can use for drug discovery. And as I was looking at your slides and seeing how the different drugs behave in the systems that you've organized, it actually is very reminiscent of what we've learned, from the clinic. And so I am excited about how your model could help us now advance all the cell immune therapies, because here you've got a mouse that actually has the immune system, it's got the tumor, it's got the microenvironment. And I was wondering if you've given thought to this and how we might be able to leverage your mouse model to help advance CAR T cells and bispecifics for treating patients with Waldenstrom's.
So this has been really a big gap in the field because Waldenstrom's patients are desperately waiting for some kind of immunotherapy. And as you said, Steve, not hand-me-down immunotherapy, but something specific to this disease. It's a very unique disease because in the bone marrow, you got plasma cells, plasmacytic cells, lymphocytes. They all have some common antigens, but they also don't have some kind of antigen. So let's say plasma cells don't have CD20, which is a target in many B-cell lymphomas. And so we can't really study those therapies before clinical trials in vitro because in vitro, the plasticware doesn't have any immune system. The mouse has an immune system. And so all the mice that I'm studying, they have fully functional immune system. And we do hope that developing some kind of CAR T-cell therapy that would target something present on all the components of Waldenstrom is definitely something that can be done.
Very exciting.
It's great. Thank you. I just want to tell everybody, because there's a little secret here. During Philip's presentation, Steve leaned over to me and he said, I still don't understand that with all the stuff about the mouse model. All I know is it works. So everybody can feel better about not understanding all those maps we had. So thank you very much.
Sure.
Very exciting. Transgenic mice are really exciting, but I think when you can utilize them to study the disease pathogenesis, but also help us advance therapies, it really is impactful. And so when we're thinking about investing in models, this is actually a really important model for us to invest. So I think we need to move on just because I know we have a break time coming up, but we have some questions for David now. And do you want to start?
No.
No, you want me to start? Okay. Okay. David, I'm excited because you have really done a lot in the myeloma space, understanding precursor conditions and the evolution to symptomatic disease. You had also a very beautiful review in Nature Review Genetics recently. And what I learned from that was how important the microenvironment, particularly the immune microenvironment, other immune cells, how they're duped by the Waldenstrom cells to allow, growth of the Waldenstrom's tumor cells. Can you tell us from your myeloma work that you've done, how you hope to leverage what you're now understanding about Waldenstrom's, and how much overlap it really is there between myeloma and Waldenstrom's?
Yeah, that's a really great question. And I think this is something that Dr. Konishi and Romanos from our lab, Sclavelitis Pistophilis, kind of worked on. It goes in the direction of also what David Moreno just showed, right? So the question is, what can you use? You can do these single cell sequencing to look at how is the bone marrow actually, what is the composition of the bone marrow, looking at different immune cells, comparing smoldering myeloma, smoldering Waldenstrom's. And it seems like when you look at these immune cells that they are kind of immune suppressed. So the immune cells which should normally target the Waldenstrom cells and kind of kill them and see them, it seems like they're changed and systematically changed in the body in a way that they do not recognize, or they're too tired and exhausted to attack the Waldenstrom cells. So this is also very exciting to see this in the single cell and really define what does these cells look like phenotypically or functionally. But I think this that's why we go into the spatial direction, right? Because like we wonder if this is true for all of the immune cells across the entire bone marrow, or is this only true for the ones that are like sitting next to the Waldenstrom cells? So I think this is going to be like an interesting question to see. Yeah.
Your lab has put a lot of effort into understanding these dynamics in myeloma. Do you see much overlap in mechanistically in how Waldenstrom's evolves to that of how myeloma evolves?
I think there is an overlap. Yeah, there is an overlap. The cells are different though, right? So myeloma cells do have like different mutations. They have a different pathogenesis. So the way that they become cancer cells is just different than for Waldenstrom cells. So they do have different effects on the surrounding cells as well. But for sure, there are overlapping mechanisms. Yeah.
Oh,
Thanks. Thank you very much. Tina, how do you
Yeah.
like having your picture in front of 200 people about 18 feet by 20 feet? I'm sorry we've had to do that to you. Anyway, thank you very much for your presentation.
Thank you.
I found it intriguing. I have just a long-term question. What do you have to have, or what could the IWMF perhaps provide for you to continue your focus on WM and not leave our little orphan disease and go elsewhere? So what is it you need in the future to continue your studies and your understanding of WM?
Yeah, well, great question. To be honest, I'm coming from a myeloma field as well. So I've been working in myeloma until 2017. And then I started gradually working in Waldenstrom. And really, I had no idea about Waldenstrom before that time. But it actually really actually captured my interest because it has so unique biology compared to myeloma. And it feels like there are so many things to be identified, studied, and I feel that, to be honest, that as I'm doing more of these studies, like I'm working on the clonal evolution of IgM Gast Waldenstrom, and now we're working on this ibrutinib resistant mechanism, and as I'm getting more and more data out of these studies, it feels like there are so many other unanswered questions. So I feel like there is a long way to actually, for me, to work in Waldenstrom, because I find that there are so many questions to be answered, and these actually are really of a great interest to me. So hopefully I'm going to stay at Waldenstrom for a while, hopefully.
Okay, we will have a 2025 RFP, and we will be awarding the results from the best in 2026. So I'm hoping that Tina will combine with some people and make an application for one of the IWMF research grants for 2026, so we can continue to have you at our ed forums and contributing. So
Thank you
if
so
you would
much.
keep that
Thank
in
you.
mind,
I'm going to try my best.
and when you see the RFP, look at it, okay? Thank you very
Yes,
much.
we already know that. Yeah.
Tina, Steve Trion here. We're very excited about the work that you're doing, and I think really for our audience, it's important for them to recognize that you're going to try to give us a sense of why it is that patients who go on drugs like ibrutinib have success, and why in some cases then you see progression of disease. Do you think these insights, because you're studying ibrutinib patients at this point, do you think these insights will also translate over to patients receiving the other BTK inhibitors like zanubrutinib as an example?
Yes, to be honest, we have a small study at the moment that we are actually studying ibrutinib and zanubrutinib in parallel with BCL2 inhibitors. This is venetoclax. And we are trying to do these studies in Waldenstrom cell lines, as well as a small part in mice. And we can actually see the differences there. There's some very interesting results. So how zanubrutinib works much better from ibrutinib in many ways, and how it targets more pathways And we are actually very, very excited about these results. And we are actually trying to put everything together. This is a small study, but it actually came up to be very, very interesting because we actually had the access to zanubrutinib and all of these mice. And we're trying to actually create some mutagenesis parts in it as well with CXCR4 mutations and see how ibrutinib is effective compared to zanubrutinib and vice versa. So yeah, it is very, very interesting field. And I think that all of these second and third generation BTKIs are going to work in much better way.
Wonderful.
Thank you very much, Tina. Thank you both for your contribution. Thank you for moderating this, and it was wonderful. So thanks, guys.
Yeah, I think we need to give a loud round of applause to our future leaders in Waldenstrom. So thank you so much for being here. Tina, thank you. David, wherever you are, thank you as well.